My name is Amber. I'm on the EU AIMS communication team. I'm delighted to introduce Dr. Eva Loth. Eva is the Sackler Lecturer in Translational Neurodevelopment and the EU AIMS Project Science Coordination and Deputy Lead Clinical Research. Eva will be giving an overview of the EU AIMS Longitudinal European Autism Project, also called LEAP, and she will explain what biomarkers are and highlight the need for biomarker research in autism. Thank you very much, Eva, and I'm looking forward to your presentation.
Thank you very much, Amber, for the kind introduction, and thank you very much to all of you who joined our second webinar. I believe we have about half an hour for this presentation and then about another half an hour for a question, and I'll try my best to answer them. So some of the most common questions that parents have when they first learn that their child has a diagnosis of autism is, what is the prognosis of my child? How will my son or daughter be when they become grown-ups? What are his or her treatment options, and what might have caused his or her autism? Now, these are terribly hard questions, and as much as they are common and obvious, we don't really have great answers to them as a field. But we do know a couple of things. In terms of the prognosis, there was a very nice paper published in 2012 that showed rather distinct developmental trajectories in terms of the social communicative symptom development as well as in the development of repetitive behaviors between the ages of 3 and about 14 years. So the prognosis was very variable. Some children started off having very severe symptoms at the age of 3 years and also had relatively severe symptoms as they became adolescents. But there was also a subgroup of children that made very significant gains. So one really important question, an intriguing question, I think, is why is it that some children make these very significant gains but others do not? And why is it also that some children develop common comorbidities, so for example ADHD symptoms as they enter school, or epilepsy, or later on anxiety or depression, but others do not? We do believe that level of intelligence and perhaps notably language abilities are the best predictors of adult outcome. But beyond that, we don't really know whether these different symptom trajectories might reflect differences in the underlying biology. In terms of the treatment options, at the moment we have no medical treatments for the core symptoms of autism, so for social communicative impairments or the range of repetitive and restricted behaviors and interests. There are a number of different behavioral interventions, such as applied behavior analyses or various targeted cognitive therapies. Some of them start already very early in development and have reported quite significant successes. But probably it also is fair to say that a lot of them are quite time-consuming, often requiring several weekly sessions. And on the whole, one of the challenges is that even though children or people who receive these treatments might benefit from them very much in terms of their behavior in that targeted treatment context or intervention context, but a common difficulty is to generalize skills that were learned in that setting to the real life. We do have also a number of different medical treatments for associated and comorbid symptoms, but again, no treatments for the core symptoms. In terms of the causes of autism, until recently, we thought that autism was idiopathic, and that was arguably also one of the reasons why we have so few or no medical treatments. With idiopathic, I mean the causes were unknown. But about 10 or 15 years ago, this has changed as common as well as very rare genetic risk factors have been identified. Now these are hundreds or perhaps even thousands of different common or genetic or rare genetic risk factors, but also a range of environmental risk factors, notably some that might have an impact during the mother's pregnancy, have been identified. Together, they might explain the causes of autism in about 10 to 20% of individuals with autism. That's quite a moving target as more and more risk factors are being identified. Now, these rare genetic forms of autism or monogenic forms of autism have had a particular significance because they seem to provide a new window to identify a causal link from a gene to perhaps underpinning cellular or molecular mechanisms that might cause some of the autism symptoms. Another important sort of piece in the puzzle was the discovery that even though we think that there are hundreds of thousands of genes involved, they might actually converge on a much smaller number of common pathways, perhaps at the molecular or cellular or brain systems level. And this is significant because it means that perhaps treatments that might be identified using this genetic approach might not only be useful for people who have this particular genetic abnormality, but also for somewhat larger patient groups. Okay, to summarize some of the challenges for the development of treatments for autism is the enormous clinical and etiological diversity, that is, diversity in terms of the likely origins. Also, different people with autism have different treatment needs. So perhaps some people with autism might want or need treatments for some social difficulties, others for particular aspects of their repetitive behaviors, another person for perhaps social anxiety. But it's also likely that very similar symptoms, for example, a particular difficulty in making eye contact, might have different neurobiological causes in different people with autism. And so these sort of challenges and questions that I highlighted have in a way led to the creation of the EU AIMS program. EU AIMS is the partnership between several academic institutions across Europe, as well as industry partners and partners from autism charities. And what we've been trying to do is to, under one roof, have 5 integrated programs ranging from cellular assays, animal models, translational science, clinical research, and genomics in order to pursue what we call precision medicine of autism. Now, what does precision medicine mean? It basically is assuming that there might not be a one-size-fits-all treatment approach for autism, whether this is medical or non-medical, but that different people with autism might need different types of treatments. So precision medicine is based on 2 pillars. One is to use animal models and cellular assays to identify new treatment targets based on the underlying pathophysiological mechanisms, not only the symptoms. And perhaps there will be other webinars in the near future that focus on these animal models and findings from cellular assays. But today, I will be primarily focusing on the identification of stratification biomarkers, and this is important for one reason, to select patients for a particular treatment. Now, what are biomarkers, or short for biological marker? It can be defined as a kind of objective test or measure that indicates a typical or atypical biological process or the response to a particular treatment. This is a fairly abstract definition, and I'm trying to make this a little more concrete in the next couple of slides. But perhaps the main emphasis here of this initial definition is the word objective. So something that really can be tested and measured regardless of whether we are in Europe or in America or in Asia or anywhere else in the world. It could be, in principle, any kind of test ranging from a genetic test, blood test, a brain imaging scan, an EEG signature, or a cognitive or a psychological test. Now, what is important is that there are different types of biomarkers. And perhaps most people, when they think about a biomarker, they think about a diagnostic biomarker. And that would be a kind of test that would help us to diagnose autism more reliably. So in this cartoon, you would have a test that would say, well, everybody with autism fulfills a particular criterion, and that is rather distinct from people who do not have autism. And so it is based on the assumption that there might be a biological characteristic that is shared among all or most people with autism. Now, whether or not this assumption is true, we do not know quite as yet, and hopefully some of the research that we're doing in our longitudinal European autism study will help us to address this. But I highlighted early on in my talk the diversity of people with autism. So there is maybe reason to believe that maybe not everybody with autism might share any particular characteristic. So here we then move to the second type of biomarker, a stratification biomarker, and its main goal is to subdivide people with this very heterogeneous condition or diverse condition into different subtypes of autism that might be more biologically homogeneous. And a stratification biomarker might serve different purposes. It could be a risk biomarker that might help to identify autism even before the behavioral symptoms manifest. And we in EUAIMS are trying to address this by a second longitudinal study that I'm not going to talk about much today, which is called EuroSIPS and which is led by Mark Johnson and Emily Jones. A second purpose of a biomarker would be to have a prognostic value, and that is to predict indeed the natural course of a particular person with autism over development. In other words, to try to address the puzzle that I started off with at the beginning of my presentation. And then a third emphasis of or potential value of a stratification biomarker could be that it is predictive. And by that I mean that it helps to predict a person's response to a particular treatment or intervention. So at the moment we have exactly 0 validated stratification biomarkers for autism. Why is this? So perhaps on the one hand, it's because most studies, in particular neuroimaging studies or cognitive studies, include quite small and heterogeneous samples. It is not uncommon, for example, in neurocognitive research to have case control studies with 20 people with autism that are compared to 20 controls. One could argue that some of these studies are already limited in terms of their statistical power to even identify a case control difference, but this is very likely true if one wanted to subdivide that sample into 2 or 3 or more subgroups. Then also different researchers and different study groups often use different experimental measures, including different versions of a cognitive test or a different neuroimaging protocol. And so because of that, we are at the moment faced with a problem that is called failure to replicate, that different studies produce very different outcomes, and we do not know whether this is due to methodological differences between the different studies or maybe indicative of real subgroups. And so therefore, one of the things that I would like to advocate for is to move from declaration of mean group differences to begin to better understand individual differences. And this is also what underpins our longitudinal European Autism Project, which is a large study that is carried out simultaneously in 7 study centers across Europe, here at King's College London, at the University of Cambridge, at the University in Nijmegen, in Utrecht, the Central Institute of Mental Health in Mannheim in Germany, and the Biomedical Center in Rome. In addition, we have a very special group of twins that are discordant of autism and are seen at the Karolinska Institute in Stockholm in Sweden. So we used very lenient participant inclusion criteria. We included participants between the ages of 6 to 30 years with an existing diagnosis of autism based on either DSM-4 or DSM-5 criteria. We allowed all comorbidities except for psychosis or bipolar disorder, recognizing that it's up to 70% of people with autism who do have one or more comorbid symptoms or conditions. We also allowed people on stable medication, again because the real world indicates that it is in different countries between, again, 30 and 70% of people with autism who do receive some kind of medication for comorbid symptoms, for example, sleep abnormalities or behavioral problems or others. Also, and this is fairly unique, I think, is we included a group of people with autism who have also mild intellectual disability, and that is defined by IQ between about 50 and 70. And this addresses a bit of a vacuum because most of our clinical research studies over the last decade or so primarily focused on high-functioning people with autism who also have IQ in the normal range, whereas 50% or more of people with autism actually do have some form of learning disability, mild, severe, or even profound. Now, we have a very ambitious assessment protocol. So, and that includes parents, or when there are high-functioning adolescents or adults with autism, the person themselves usually come and complete a battery of online questionnaires at home that they can log into. And we are asking for autism symptoms, comorbidities, quality of life, but also about environmental risk factors, as well as information about the medical history. Then the person with autism and one parent come and visit us on 2 separate occasions. And I've just given some examples here. So, for example, they come and have a comprehensive clinical assessment, including the ADOS and the ADI, to verify diagnosis. On another day, we also ask parents to give information about their son or daughter's adaptive behavior. We have a range of cognitive tests. Some of them use eye tracking. We assess brain structure and function using MRI and EEG. And we test blood and we take blood and saliva samples for biochemistry or genomics. So this slide gives an overview of our baseline cohort without the twins, and we have now assessed with this comprehensive battery over 700 people, including 437 people with autism and about 300 controls, that is people with typical development or mild intellectual disabilities without autism. And this is just an overview of our recruitment graph. So we started with our assessments in February 2014 and completed the baseline assessment about February 2016, so in about 2 years, except for Schedule D, and these are the people who also have mild intellectual disabilities. So these are adolescents or adults with also mild intellectual disabilities, with or without autism. They were a little harder to recruit, and so therefore we decided to continue with recruitment up until now. But since February 2016, we've also started with our first follow-up assessments that included pretty much the same structure as our baseline assessments and most of the very same tests. and we have, as of the end of June, seen about 500 people. So I will not go into detail of the next slides, but suffice to say that we had very good acquisition rates across the different modalities, such as cognition, eye tracking, most notably MRI scans as well. EEG rates were a little lower because one site didn't include EEG, as well as the blood samples. So one question that some of our parents are asking when they're coming back to our follow-up assessments is, why don't we have so many results as yet? Or why haven't we learned about our results from the baseline visits as yet? And the reason for that is that it took us quite a long time, and this is still ongoing work, to analyze our data. And that starts with the data cleaning, or so-called quality control procedures. So, for example, for our MRI brain scans, there is a dedicated team that looks at each and every one of these different brain scans, so for structural sequences, for diffusion tensor imaging, and for the functional sequence, to see whether they have good quality or not. So this is an overview of our analysis plan. So what we want to do across different data modalities, and with that I mean the cognitive tests, the eye tracking, the EEG examinations, and so on, we first start to look at case control mean group differences. And then we want to subgroup by factors that we believe or hypothesize are plausibly affecting biology or cognition based on previous work. So, for example, it is now well known that there are differences between males and females with autism, and Meng Chuan Lai will talk about this specifically in the next webinar. It's also very likely that there will be developmental or age differences, given that autism is a neurodevelopmental condition. There might be differences between the people who have common comorbidities and those who do not have these comorbidities, or who have differences in terms of their IQ or have mild intellectual disability. Then we want to see also how group differences relate to symptomatology. And then what I listed here as the fourth point, we also begin to use more novel, data-driven, multivariate approaches to identify subgroups. And I want to give you an example from our cognitive analysis so far. And the reason why I chose that is because we have made relatively more headway with that than with some of the other analyses, because they didn't require quite so much quality control procedures as, for example, the structural functional neuroimaging data. We have a battery of cognitive tests that are all tests that have been previously used in autism research, are quite well known. Tapping, for example, what we call theory of mind, the ability to understand other people's thoughts and emotions and feelings or intentions. We use from our eye tracking battery indices of spontaneous social attention to social aspects of a scene or non-social aspects. We have a task of biomotion, again administered with eye tracking, to tap into the very early occurring typical tendency to find biological motion more interesting than other types of motion. And then we have also a number of what we can call domain general cognitive tests, tapping, for example, spatial working memory or cognitive flexibility like reversal learning, or a test of weak central coherence, the ability or the idea that some people with autism have a more detailed, focused processing style, perhaps at the expense of integrating information in context or for meaning. So this is just a summary of the individual case control differences or case control comparisons, and what you can see here is that on the whole, for most of these tasks, we do replicate significant mean group differences. We have higher p-values than you might be used to in previous studies, which is simply to do because we have such a large participant group. But the effect sizes on the whole were relatively moderate. this highlights this from one of these tests, the spatial working memory test, where we have a significant case control difference, and the traditional way of interpreting these findings or concluding is that people with autism have, on average, significant deficits in spatial working memory compared to controls. But what does that mean for individual people with autism? And arguably, if you have a child with autism, or you have autism, you might not only want to know what people with autism have different on average to people who do not have autism, but you want to know what that means for yourself or for your child. And to begin to address that, we use a technique called normative modeling, and that's a little bit akin to deriving growth curves, as you may be familiar with, for example, for height and weight. So what we did here is we plotted the development of task performance scores of the typical developing group from childhood through to adulthood, and also the standard deviations for each different age. And then we could plot here how far each person with autism deviates from the age expected mean. And what this showed was a rather different finding, namely that it was over half of the people with autism who actually performed right within the normal range or even above relative to their age. And only about 30% of people with autism showed what are likely clinically relevant deficits, that is to say, deficits more than 2 standard deviations below the typically developing mean. And I think this is very important to bear in mind, in particular in relation to the conclusion that are typically drawn from the mean case control differences. Now, the next question that we were asking was, well, it could be that performance on a single task might not be so helpful to predict how they affect everyday life or symptom severity. Might it not be more useful to consider somebody's cognitive profile, strengths and weaknesses across the different cognitive domains? And in order to begin to explore that, we entered all of our cognitive, or most of the cognitive measures that I have told you about before in what is called a hierarchical clustering analysis, and have found 3 distinct clusters or subgroups of people with autism. One that could be characterized by showing intact theory of mind, but problems with either executive function or reverse learning or spatial working memory. Another, perhaps astonishingly large cluster, were people with autism who actually had no differences across the board. And then we found a larger third cluster of people who had impairments both in theory of mind and executive function. And what was then quite interesting was to see how did these 3 subgroups differ in terms of their clinical profile. And what we found was that the cluster 3, who had impairments in both theory of mind and executive function, also had more autism symptoms both in the social, communicative, and repetitive behavior domains in the 2 other clusters. What was interesting was that the cluster of the people who were good across the board, they weren't better in terms of their symptom severity than the cluster who had intact theory of mind but impaired executive function. But it seems that their intact cognitive abilities helped them to have significantly better on average adaptive behavior. So in other words, they were faring better in adapting to the demands of everyday life. And also interestingly, they had on average fewer comorbid symptoms, both in terms of ADHD symptoms and anxiety and depression, than the 2 other subgroups or clusters. Okay, I'm coming almost to the end. So the next question that one could ask is, so that might be interesting. How can we use these kind of findings in clinical practice? And so this is where I have to be a bit more cautious, because we are excited about these sort of findings, but that's really only the very beginning of a still long way to go. And this is indicated here by this first arrow indicating the possible identification of a candidate stratification biomarker. That's in principle a great start, but we really must not stop here. So the next step would be to validate that stratification biomarker. And that means to say, is this test actually reliable? Is it measuring reliably the same, for example, if we gave this test to this person on Monday morning or on Friday evening? And if there are differences, do we have to account for that, for example, whether it's the morning or the evening, or fatigue or something? And then the next really important thing is we need to reproduce our findings in a separate cohort, and also have to ascertain whether it is really sensitive and specific, our subgroups. Once we have done that, or in parallel rather, the next step is, or the actually parallel steps, is to qualify the biomarker. So next we need to find out, do our different subgroups, cognitive subgroups here, have different etiologies, different symptom profiles? Yes, we are indicating that preliminarily. Different developmental trajectories, we don't know yet. Or do they differ in terms of their response to a treatment, medical, or intervention? And in order to do that, to test that, we would need to include these tests in a treatment study. If we get confirmation from that, the next step would then be to go to a regulatory authority in Europe, the European Medicines Agency, or in America, the FDA, and see whether they would approve our candidate or our biomarker. And once that is done, we can think about how we could implement that in the clinic. And for that, it's very important to know that the marker is safe and easy to measure. Ideally, it would be also cost efficient, so not too expensive. And then we need to find out whether clinicians find the marker useful, or patients, and whether it really makes a difference in real life. So to conclude, why do we need stratification biomarkers? So we hope that validated stratification markers will be useful for clinicians, families, and researchers to better understand what causes the core symptoms of autism and also the associated symptoms of a particular individual with autism, to aid in more reliable diagnosis by knowing what kind of subgroup or subtype of autism somebody might have, to help predict the natural course of somebody's symptom progression over development, to aid in the development of medical treatments, but then also to predict how different people with autism might respond to any given treatment or intervention, whether it could be useful for this particular person. And if we know that, then it's also much easier for parents and for people with autism to take informed decisions as to whether or not they might want a particular treatment. And so my take-home message is that a significant group difference that most of us have been focusing on in case control studies doesn't really make a diagnostic or stratification biomarker, and that a biomarker is always making some kind of predictions for a particular individual. So thank you very much for listening. Our next ERAIMS webinar will be held by Dr. Ming-Chuan Lai, and it will be on why are sex and gender important for understanding autism. And I'd like to close by thanking all my colleagues at the various different institutions that together really have produced the work that I've presented. Thank you very much.
We've received some questions in advance of the talk. So if there are no questions from the audience at this time, we will first address the previously submitted questions. Okay, so Eva, if you do find validated biomarkers, for example, in fMRI or in genetics or in blood, could you see a time when this will move diagnosis and identification away from behavioral and clinical observation?
So it's very difficult to have a particular timeline. Perhaps the best I can do at the moment really is to remind you of the different steps that really are needed to go from identification, for example, of a genetic test or a brain scan that might be indicative of different subgroups, to then the validation, qualification, approval, and then fit for purpose validation of a biomarker. If I really had to say something, I would imagine it would be at least 10 years or so from the moment that we have identified something.
If you do identify subgroups, what is the next step in developing a targeted treatment for that group? So you're saying we have to go through all these motions, but what specifically would you be addressing?
So, in a way, the biomarker work is quite closely intertwined with the parallel work of identifying new treatment targets. So because for a biomarker to be useful to predict the response to a particular treatment, it has to be matched to that treatment. So this is another challenge. But maybe I can give you an example, a more concrete example of how maybe a biomarker could be useful. So, and this is really hypothetical. So, some people with autism, we think, have difficulties with making eye contact. And so there are at the moment two separate hypotheses of what might underpin that. One would be that maybe some people with autism have what is called diminished social reward or diminished social motivation. Perhaps people or eyes might not be quite so salient for this person with autism. Another hypothesis is that maybe some people with autism have more social anxiety, so they might avert their gaze because that is too overpowering. So it could be the same behavior, but has very different neurobiological or cognitive underpinning, and might therefore also require different treatments. And if you knew what might be the case for a particular person, then that might be useful for a clinician to decide, we want to treat more for social anxiety or more for social reward.
Very interesting. Again, if there are any questions from the audience, please feel free to post these in the public chat or use the Q&A button at the top. I have some general questions. What could projects like EU AIMS and LEAP mean to people with autism and their families in real terms? So the kinds of challenges they face on a daily basis.
It really is a bit sort of looking out into the future. I think that we do this because we assume that we have a chance to find a stratification biomarker. And if we were to do that, then I think that could really have a tremendous impact on people's lives. Because it would help them to steer them towards potentially different treatment options. It would help people to understand, what is the likelihood of prognosis of a child, and to be prepared of different, developmental milestones and so on. So it might inform what kind of school would be more appropriate for one child or another child, and so on.
Great. And does that also mean that, for example, special educators can be trained in recognizing biomarkers or understanding biomarkers to really integrate this in the school system?
Yes, I think that's a great idea. And again, I think one of the things that we can already say is really that autism is very diverse, and so that, for example, from the perspective of someone in the educational field, that really something that might be very useful for one child with autism might not be quite so useful for another child with autism.
And then I have another general question.
Mm-hmm.
Could you tell us more about how the project is looking at the co-occurring difficulties that are often present for those with autism, like mental health issues or other neurodevelopmental conditions?
Yes, so we were sort of like preparing for that by including, in terms of our participant inclusion criteria, people who have any kind of comorbid symptom. And so what we are now trying to do is to maybe a priori subdivide our sample by saying we want to compare the cognitive profile or the brain imaging, functional imaging scans of people who have autism and no comorbidity to people with autism who also have anxiety or who also have depression, and see whether they have similar or differences in terms of, for example, their responses to emotions, either on a behavioral basis or on a neurofunctional basis.
Yeah. Okay. Are there any other questions from the audience? If there are no other questions, I would like to thank Eva again for a fantastic talk, and obviously also thank you to all our participants for participating. And we look forward to seeing you at our next webinar, which will be on Wednesday, 27th of September. So we're taking a break for August. And we hope that you will join us again in September. Thank you, Eva.
Thank you very much, Amber, and thank you very much to all of you for listening to this webinar.